Welcome to America's Web Radio. This is Ron Bachman, and you're listening to Healthcare Insight. Well, once again, COVID is in the news because of a new variant, Omicron, and that follows up on the Delta. And lots of controversy now, uncertainty about what this coronavirus variant mutation represents. Seems to have started someplace in Africa, out of the southern part of Africa, and it's spreading itself across the world. It's supposedly, according to the experts, very contagious. So there's a possibility this is going to spread throughout the world very rapidly with some unknown consequences. The original doctor who identified this mutation of coronavirus says that it is very contagious, but that the symptoms are very mild. And as a result, if that's true, other doctors have said this could be a real saving grace and keep us away from the disasters that we've seen in the past from the original coronavirus and then the Delta variant. Because people would get immunities with very little side effects, and it could actually be the way that we overcome the worldwide pandemic. On the other hand, if that doctor who originally discovered this variant in South Africa is wrong, and there are more consequential issues of getting sick and hospitalized and death from this variant, it could make things much worse. And so right now there's a great deal of uncertainty. The stock market is dropping like a rock on several days here because it could affect the economy, could affect worldwide distribution of goods, our supply chains that have been problematic in getting goods to the marketplace could be even more affected. The workforce across the world could be affected again. So there's a lot of uncertainty going on. So I wanted to come back and talk about the coronavirus issue itself. Many of you have kept up with some of the stories and backgrounds and the criticisms of Dr. Fauci, the criticisms that have been made by Dr. Rand Paul, and the studies that have been uncertain, unclear, confusing as to whether this thing actually was originated in Wuhan or not. I want to bring that to closure and give you information that's been out there for a long time that anybody can really read, but I've discovered it in real journalistic, scientific studies about what the true story is and what's the likelihood that it really was developed in Wuhan. So I want to talk about coronavirus. I hope you're not sick of listening to this stuff, but I think you're going to hear more stuff today that you've not heard before and put together in a clear understanding, because this thing doesn't seem to be going away. Maybe this new variant will allow us to really minimize the impact of the pandemic, but it's something we are all living with now for over 2 years. And if something doesn't happen, if this isn't a final sort of push, because there is a history of mutations of viruses getting weaker and weaker as they mutate, but they never fully go away. So we're probably going to have to learn how to live with some version of this, just like we do with the annual flu. We'll probably be getting annual COVID shots, and hopefully nothing else like this ever erupts from the scientific manipulation of viruses. So let's continue with this discussion and find out what really happens worldwide with this new variant and with the understanding of where the coronavirus actually started, how it came about, and help us to understand the language of the coronavirus and the epidemiologists that are working on this, the virologists that work on this. Now, the key question many in this audience might be asking is, well, I've been vaccinated, so am I safe from this new Omicron variant, this mutation, or am I not? Am I partially protected? Is the protection no longer 95%? Is it 60%? Is it 40%? And if I get it, am I likely to be hospitalized? What's the risk of death under this? And there's a lot of uncertainty about that. The University of Oxford said this past Tuesday that there's no evidence that the current COVID-19 vaccines would not continue to protect against severe disease from the Omicron variant. So again, it kind of matches up with what the original discovery in South Africa was all about, and that it is very contagious, but it is not deadly. I want to take today and really get into a clear understanding what this coronavirus is all about. And as we start that process, we need to have some understanding of terminology. As laymen, the scientific language doesn't always resonate, or we don't really understand what it is. But let me start off with two definitions that are really basic to the understanding and the development of the research that I've done on this whole issue. The first is that a disease and a virus are two different terms for the same thing. The disease, for example, the coronavirus disease that we're currently calling COVID-19, that is a short form of really the coronavirus disease dash 19. And then there's the name of the virus itself. So the disease is coronavirus or COVID-19, but the virus that causes that disease is called something else entirely. And the name of the virus is SARS-CoV-2. And SARS stands for Severe Acute Respiratory Syndrome. A basic question here again is, why does the virus and the disease have different names, and who names them? Well, viruses and the diseases that they cause often have different names. For example, HIV is the virus that causes AIDS. So people often know the name of the disease, but not the name of the virus that causes it. But most of us know HIV, AIDS. We kind of throw all that together, but HIV is the virus and AIDS is the disease. So the World Health Organization announced COVID-19 as the name of the new disease on the 11th of February 2020, following guidelines previously developed with the World Health Organization for Animal Health and the Food and Agriculture Organization of the United Nations. So technically, the disease is a coronavirus disease, but the underlying virus is a SARS coronavirus. So I hope that clarifies two important terms. So people around the world commonly get affected with human coronaviruses, but they're typically that common form in the first four categories. I didn't get into a lot of details, but they're not particularly harmful. They don't kill a lot of people, may kill some people, but they're nothing like the pandemic potential of these last three, the MERS, the SARS-CoV, and the SARS-CoV-2. And remember, we're talking about SARS-CoV-2 in today's environment with this COVID-19. So there are currently 7 coronaviruses that are known to cause disease in people, but so far, only those ones I mentioned have caused large outbreaks and are fatal and have fatal illnesses in people. SARS was first reported in Asia in February 2003, and over the next few months, the illness spread to more than 24 countries in North America, South America, Europe, and Asia before the SARS global outbreak in 2003 was contained. Bats have been identified as the natural resource of severe acute respiratory syndrome, or the SARS-like and SARS coronaviruses. The discovery of bat SARS-like coronaviruses and the great genetic diversity of coronavirus in bats have shed new light on the origin and the transmission of SARS coronaviruses. And that's what we're going to dig into the rest of this hour, the source and the transmission of these SARS coronaviruses. But keep in mind that we actually have in 2021 more deaths identified with the COVID disease than we had in 2020. Most people don't know that because the media doesn't sort of promote that, but we do have a lot of people still dying who mainly are unvaccinated. So the other question would be, if I'm not vaccinated, am I likely to get this and have the same kind of severity or disease or hospitalization rates or even death rates that people have seen before? And again, the answer is somewhat uncertain, unclear at this point. We'll figure that out over the next few weeks or months. But it does appear that this could be a good thing with people getting the antibodies, but not getting sick if that is the nature of this particular Omicron variant. But time will tell. So let's continue with the process of understanding the core issues around the COVID issue and also these multiple variants that seem to be developing. And keep in mind that historically, at least, the mutations tend to be weaker and weaker over time, and we hope that that's what's actually happening here. Well, in the last few minutes of this first segment, let me define a really key term here. So for laymen, there are two important terms, but I think I'm only going to have time to really define the first one. Then we'll come back in the next section and I'll define the second one. The first one is called gain of function research. And what is it? It's a line of research where scientists take viruses and study how they might be modified to become deadlier and more transmittable among humans. Why would they do this? Scientists who engage in such research say it helps them to figure out which viruses threaten people so they can design countermeasures. Now keep in mind that viruses mutate in nature. Most viruses and animals do not mutate to create human transmissions. So this research tries to anticipate potential mutations that could affect humans. By doing so, they can develop potential cures for when such natural mutations occur. So recognize that these mutations occur all the time. And now we're seeing more and more mutations occur. And the latest one is Omicron, hopefully is one of those mutations that will occur that says that the virus is kind of dying off. It's trying to stay alive in the host, but there's so many vaccinated people out there and people who have had COVID that have natural immunity that is trying to find a way to spread itself, even if it is less deadly to the host it's going into. So that's what we're seeing, hopefully, in this Omicron, but we will find out in the next few segments of this program how all this is playing out. Let me stop there and we'll come back and we'll talk a little bit more about this gain of function process and why it's so important and why it's so dangerous. So let's take a quick break and we'll be right back here on America's Web Radio. Welcome back to America's Web Radio. This is Ron Bachman and you're listening to Healthcare Insight. The first segment of this hour, we talked about some definitions of the virus, the coronavirus, the COVID-19, and why it's also a SARS virus. In this session, what I want to talk about now are some of the scientific names, if you will, of what's going on, the things that they do, because these are terms that as a layman I didn't know existed. The first one we defined right at the end of the first segment. It's very important, so I want to remind you, it's called gain of function research. And it's how scientists will take and manipulate viruses and study how they might be modified to become deadlier and more transmittable. Now, that sounds like a very dangerous thing to do, and you wonder why they would do it. Well, they do it because viruses mutate in nature, and so what they're trying to do in the lab is replicate some of that changing function that will occur during mutations, known as the viruses evolve from one state to another one. They're what's called mutating. And most viruses, when they mutate in nature, really don't mutate to become transmittable to humans. Sometimes they do. And so this research tries to anticipate potential mutations that could affect humans. And by doing so, then they can take that virus and try to develop cures. But until they develop a cure, you have these newly developed lab clinical research viruses that are very dangerous. And that danger is not without being extreme if it's not being performed in 100% secure research labs. I mean, developing lethal viruses that do not exist in nature exposes really significant human-to-human transmissions that would create a pandemic. So it has to be in a research lab that is 100% secure. Well, guess what? They're not always very secure. This is an example. Back in 2014, the US government had placed a federal moratorium on this process called gain of function, or GOF research. It put a moratorium on the funding for altering natural pathogens. Again, that word that we described earlier, which just means it's a virus that transmits diseases. So altering these natural pathogens or viruses to make them more deadly and infectious as a result of rising fears about a possible pandemic caused by an accidental or deliberate release of these genetically engineered monster germs. There was great fear in 2014, and so a moratorium was placed on that. Why? Well, in part it was due to a number of lab accidents at the U.S. Centers for Disease Control and Prevention in July of 2014 that raised questions about biosafety. At the time, the CDC had closed 2 labs and halted some biological shipments in the wake of several incidences in which highly pathogenic microbes were mishandled by the U.S. government laboratories. A couple of examples. In 2014, it was a very bad accident in this biohazard front. In June 2014, as many as 75 scientists at the Center for Disease Control and Prevention were exposed to anthrax due to an accidental shipment of live anthrax around the country. Number 2, there was a discovery of some forgotten live smallpox samples in a newly revealed incident in which the dangerous influenza strain was accidentally shipped from the CDC to another lab. A CDC internal report described how scientists failed to follow proper procedures to ensure samples were inactivated before they left the lab, and also found multiple other problems with operating procedures in the anthrax lab. A few weeks later, the Food and Drug Administration officials ran across 16 forgotten vials of smallpox that were in storage. Meanwhile, the largest, most severe, and most complex Ebola outbreak in history was raging across West Africa, and the first patient to be diagnosed in the United States had just been announced. That was the environment that we were working in. So it was within that context that the scientists and the biosecurity experts found themselves embroiled in a debate about this gain of function research. Was it really worth it? Well, as such, in October 2014, because of public health concerns, the U.S. government banned all federal funding on efforts to weaponize 3 viruses: influenza, the Middle East Respiratory Syndrome, or MERS, and the acute respiratory syndrome, SARS. So we banned all that in 2014. Now just keep that in mind and set that aside, how dangerous it was, how many problems with security and safety, and the potential of a pandemic if something leaked out of one of these labs that were supposed to be secure, but were not. Let's go on to a second way that scientists were working with these viruses, because it's called passaging. Again, a term as a layman I didn't know until I started to do some research on this. What is passaging? Well, passaging is the placing of a live virus into an animal or cell culture to which it is not adapted, and then, before the virus dies out, transferring it to another animal or cell of the same type. Passaging is often done iteratively. In other words, it's done one after another, after another, after another. In theory, the theory is that the virus will rapidly evolve, as viruses have high mutation rates. It changes all the time. But if they change and they become adapted to the new animal or the cell type. Passaging a virus, by allowing it to become adapted to its new situation, creates a new pathogen. The most famous experiment on passaging was conducted in a lab of a Dutch researcher. This Dutch researcher took an avian influenza flu called H5N1 that did not infect of all animals, they were using ferrets and serially passaged them to other ferrets. The intention of the experiment was to specifically to evolve what they call potential pandemic pathogens. But what happened was after 10 passages, the researchers found that the virus had indeed evolved to not only infect ferrets, but to transmit to others in neighboring cages. They had created an airborne ferret virus, a potential pandemic pathogen, and a storm in the international scientific community evolved. So think about this now. We have the gain of function that could make things more dangerous, more infectious. Now we have passaging that takes something that is not dangerous to humans, not dangerous to other animals, and passing it through this process and evolving it so that it is dangerous to others. So since these two previous coronavirus near pandemics, SARS in the 2003 and the MERS in 2012, probably both came from bats and are both thought but not proven to have transitioned to humans via intermediate animals, in particular what they call civets and dromedaries. Well, dromedaries are the camels. And the civet is a small, lean, mostly nocturnal mammal relative to tropical Asia and Africa. It's typically found in tropical forests. And the term civet applies to over a dozen different mammal species. Most of the species are found in Southeast Asia because there's a lot of experimentation going on over there in China. So this virus then evolved briefly in this animal species, but not enough to cause a civet pandemic. And then it was picked up by a human before it died out in the civecs. In this first human, so the patient zero, the virus survived, perhaps only barely, but was passed on, making the first case of human-to-human transmission. As it was successfully passed on in its first human host, the virus rapidly evolved, adapting to better infect its new host. And after such tentative transmission, the pandemic properly began. That's how pandemics occur. Scientists are manipulating viruses, creating the more serious nature of them to, in theory, prevent pandemics, but instead they're causing pandemics. So how safe is this manipulation research? Well, an accidental lab release is not merely a theoretical possibility, because in 1977, a laboratory in Russia and maybe even China were the most likely releases that, while they were developing a flu vaccine, accidentally released the extent H1N1 influenza virus. And the H1N1 then went on to become a global pandemic virus. A large portion of the global population became infected. In this case, the deaths were few because the population was over age 20, had historic immunity to the virus. But many of you may remember the term H1N1. In 1977, it got out and created this worldwide pandemic in the sense that so many people got infected, we just didn't have as many deaths because we got lucky. This episode is not widely known because only recently has this conclusion been formally acknowledged in the scientific literature and in the virology community that has been reluctant to discuss such incidents and accidents. They kind of hide things up because they don't want to lose their funding. Still, laboratories, these viruses escape, leading to human and animal deaths. Smallpox in Britain, equine encephalitis in South America are common enough that they ought to be much better known that these things have happened. Only rarely have these broken out into actual pandemics on the scale of the H1N1, and incidentally broke out again in 2009, 2010 as the swine flu. Many of you remember that, causing deaths estimated between 3,000 and 200,000 deaths worldwide. And many scientists have warned that experiments with these potential pandemic pathogens like smallpox and Ebola and influenza virus, are inherently dangerous and should not be subject and should be subject to strict limits and oversight. They should not be hidden. They should not be in labs that aren't 100% secure. So even in the limited case of the SARS-like coronaviruses, since the quelling of the original SARS outbreak in 2003, there have been at least 6 documented SARS disease outbreaks originating from research labs, including 4 in China. These outbreaks caused 13 individual infections and 1 death. Now, that doesn't sound like much in today's world, but we were just lucky that it didn't go further. But clearly, labs are not as secure as they should be. Well, the story develops. And we're going to continue the story about the current day coronavirus and the outbreak and the problems that it's caused right after this commercial. Welcome back to America's Web Radio. Well, COVID is back in the news. We've got this new Omicron virus, a mutation of the COVID that people are wondering what's going to happen. So I wanted to do this program here to go back and let people hear about the history and the background and the science of what this is all about. But I think at the end of the day, at least I'm hoping, we don't know for sure, wait on the doctors and the researchers to tell us, but I think as things mutate, at least historically, they have become less deadly, maybe more contagious, but less deadly. So I want to go back and I want to continue to talk about the origin and the issues around COVID so that we better understand this new mutation that's occurring. So we are talking about the origin, the definitions, the explanations of COVID-19. We've defined some of the terms so we have a better understanding, and we're now understanding the process of the science in researching SARS or COVID type viruses. We've learned that there is something called gain of function, where we take a virus from nature and we try to change it so that it is more deadly and more infectious. And now we've also found out that scientists use a process called passing or passaging, they use both terms, to create a virus that would go from animals to humans by having a series of mutations. So now we want to talk about how all this kind of fits together with current day. You know, as we talk about those processes that are going on in the scientific community, it's awfully dangerous, it sounds like, to laymen like myself. But I understand in talking to scientists that it is a normal way to take and develop research to find changes potentially in DNA splicing, to look for cures for things like cancers or Alzheimer's. In this case, we're using the processes around viruses, which makes it very dangerous. But the purpose is to find cures for viruses before they occur naturally in the wild and become pandemics that we know nothing about. So for these reasons, and also to ensure the effectiveness of future pandemic preparedness efforts, it's a matter of vital international importance to establish whether a laboratory escape hypothesis on the COVID-19 from China has credible evidence to support it. This must be done regardless of the problem in the U.S., of the sort of toxic partisan politics that's going on right now. We need to find out whether or not this virus escaped voluntarily or whether it was a mutation that occurred in nature. So we know how best to do future researches. So putting all this together, what's the connection to the Wuhan labs and what's been going on over there? Well, in the face of the 2014 moratorium in the United States, Dr. Anthony Fauci, the director of the National Institute of Allergy and Infectious Diseases, well, when they couldn't get the funding in the United States, when that moratorium was put on in 2014, Dr. Fauci, in running all of this work on the coronavirus, he outsourced in 2015 the gain of function research to China's Wuhan lab. and licensed that lab to continue receiving US government funding. So we funded, as a country, the Wuhan lab. Dr. Fauci's National Institute of Allergy and Infectious Diseases actually funded a study on bat coronaviruses, which was a project that included scientists at the Wuhan Institute of Virology, or WIV. We're going to talk about WIV. It's a Chinese lab. At the center of all this controversy is the one we think about when they talk about Chinese labs, but it's at the center of the controversy over this bat research. That study confirmed in 2018 that humans had died from the coronavirus. Now, one might ask, in a very serious way, if the government, the US government, banned this research in 2014 on federal funds being used for this gain-of-function research, what are the federal compliance and ethical issues surrounding the fact that the National Institute of Health still gave federal funding instead of private funding to the Wuhan lab to continue the experiments? Now, as we continue to fund work in a lab that looks like the problem started, and we'll talk about whether it was accidental or maybe some other cause. So, how did the virus escape from Wuhan if that's the case? Or is it the case? Well, in essence, the lab escape theory is what Wuhan is, that Wuhan is the site of the Wuhan Institute of Virology, the WIV. It's China's first and only biosafety level 4. That's the highest level supposed of safety and security. It's the only one in China. And so it's the highest security level of any research that can be done. But it was just added, really, while the laboratory existed for several years. The level 4 security was only added in 2018. But they had been collecting large numbers of coronaviruses from bat samples ever since the original SARS outbreak in 2002 and 2003, including collecting more in 2018. So this research was led by a Professor Shi at the WIV. The scientists also published experiments in which bat coronaviruses were included into human cells. Moreover, according to an April 14th article in the Washington Post, the US Embassy staff visited the Wuhan lab in 2018 and had grave concerns about biosecurity there. In 2018, the US Embassy in Beijing took the unusual step of repeatedly sending US science diplomats to the Wuhan Institute of Virology, this WIV, which had in 2015 become the first laboratory to achieve that higher level of security. What the US officials learned during their visits concerned them so much that they dispatched 2 diplomatic cables characterized as sensitive but unclassified back to Washington. The cables warned about safety and management weaknesses at the lab and proposed more attention and help. The first cable warns that the lab work on the bat coronaviruses and their potential human transmission represented a risk of a new SARS-like pandemic. That's exactly what happened. At that lab, it's just 8 miles from the Hunan live animal market that was initially thought to be the site of origin of the COVID-19 pandemic. That has pretty much been dismissed. Some people might hold on to that, but this does not look like it necessarily just occurred naturally in a wet market, as they called it. The Wuhan lab is also home to a lab, or Wuhan City is also home to a lab called the Wuhan Center for Disease Prevention and Control. It's sort of their Chinese CDC, if you will. Now, it's only a level 2 security lab, and it's just a short distance away from the Hunan market. And bat coronaviruses in the past have been kept at that Wuhan CDC lab. Thus, the lab escape theory is that researchers from one or both of these labs may have picked up this SARS COVID-19 like bat coronas on one of their many collecting trips, or alternatively, a virus they were studying, passaging, engineering, and otherwise manipulating escaped. Quite honestly, we don't know. I'm not trying to present some conspiracy theory. We really don't know if it was something that they picked up in nature or it happened in the lab. We know that they were doing gain of function, and we knew that they were doing passaging, and we knew that the lab was not very secure. So you can figure out what's going on here, because there is no evidence that they've been able to find. There's no other bats that in nature that seem to have this same COVID-19 type of virus. So some professors in virology at the University of Australia, since they're over there as well and have been involved with this process, they first addressed that question of whether there was a natural pathway and whether that was viable. And what they determined was that there is no natural virus matching that COVID-19 that's been found in nature, despite an extensive, intensive search to find its origins. That is to say, the idea of an animal intermediate that was working to develop this is just pure speculation. Indeed, there's no credible viral or animal host intermediaries, either in the form of a confirmed animal host or a plausible virus intermediate that has to date emerged to explain any natural transfer of the SARS to humans. In addition, there are two further difficulties with the natural transfer thesis. Apart from the weak epidemiological association between early cases and the Hunan wet market. First is that the researchers from the Wuhan lab traveled to caves in Yunnan about, maybe a mile or so away to find horseshoe bats containing the SARS-like virus. To date, the closest living relative to SARS-CoV-2 yet found comes from Yuan. Why would an outbreak of a bat virus occur in Wuhan when Yunnan is where the bats would have been? They would have affected the population there first, so there's no reason why, if it's actually in nature, that it would have occurred and started in Wuhan. And moreover, the Chinese population of 1.3 billion, if there's spillover from the wildlife trade, that was the explanation, then other things being equal, the probability of a pandemic starting in Wuhan is really less than 1% from the calculations that are made. So you can say take a bad virus that is not infectious to humans and force its selection by culturing it with cells that express what's called the human ACE2 receptor. In other words, the ability for humans to pick up this virus. The cells have been created many years ago to culture the SARS virus, and you can force this bad virus to infect human cells via mutations in its spike protein, which would have the effect of increasing the strength of its binding to humans and inevitably reducing the strength of its binding to bats. In other words, it gets transferred from bats to humans. That is the likely scenario of what's actually happened. So the viruses in prolonged culture will also develop a random mutation that does not affect its function. The result of these experiments is a virus that is highly virulent in humans, but is sufficiently different that it no longer resembles the original bat virus. Because the mutations are acquired randomly by selection, there is no signature of a human gene jockeying, but this is clearly a virus that could still be created by human intervention. So we got to make a difference between a human intervention and process that says, well, there's no evidence of human intervention. It actually can be something that the end result shows no effect of humans, but it's because of the passaging that this has occurred. Well, let's take another final break, and we'll come back and wrap this all up with where this is all going and what it might actually mean. So stay with us, and we'll be right back on Healthcare Insight. Welcome back to the final session of Healthcare Insight. Now, what have we learned today about the virus? Well, we know that after 2 more years and the federal government trying to shut down businesses, have us mask, have us get vaccinated, that mutations are occurring, and it creates a great level of concern and controversy and uncertainty about how it's going to affect our economy. We had such a disastrous shutdown of the economy. Stock market dropped 30, 40%. Everybody had to isolate themselves, cocooning themselves, not contacting family, not being able to go to church. And now we have a new variant, the Omicron variant. And the concern is, well, we're going to go back to the future again and have all these problems. I don't think that's the case, but I'm not a doctor. Everybody's got to figure it out. But this program here, this radio program, podcast, is trying to at least give us all a refresher education on the whole background of the coronavirus so that we have a better understanding individually of what's likely to happen. And the best scenario that I can throw out would be that we have a mutation that occurs all the time with viruses that will be less deadly, but maybe more contagious. And this might actually be helpful. Some scientists are telling me that I'm reading about and hearing that this would give us natural immunities, which would, in many cases, are thought to be better than the vaccine. That's another debate that can go on. But this might actually be a way to end the problems of the coronavirus from a practical and economic standpoint. On the other hand, we'll just have to wait and see. Is this going to be a real problem mutation? But I can almost assure people, just from general knowledge as a layman, that we're going to continue to have mutations, but typically, as I read it, mutations get less and less dangerous. So let's do a summary again of where we are and what we know so we can all move forward without panic, just watching the situation, living our lives as best we can, because we're all kind of sick of this, whether there's a new Omicron variation or not. We've got to move on. We can't spend the rest of our lives in our homes, isolated from family, isolated from work, isolated from the rest of the world, economic shutdown, that the problems that are being created by that are maybe worse than the virus itself, especially at this point. So let's do a summary of where we are and then wrap this program up. Well, what we've learned is that there's a difference between the disease and the virus. The disease is COVID-19, but the virus is actually SARS-CoV-2. We also found out that scientists have been working on these viruses in rather strange ways that, as a layman, we wouldn't necessarily understand, because it looks like they're making the virus, the SARS virus, worse, more profound in its impact, and also that it will transmit to a greater degree than it would have in nature. These two things, this gain of function and this passaging, are enormous insights as to what the scientific community has been doing to manipulate the virus, to create a more virulent virus. And we can see that they haven't been very safe in these labs that are supposed to be safe. The things have gotten out. There have been misplacements of the viruses. There's been a forgetfulness about where the viruses were stored. There's a sloppiness to the scientist community working in these labs when it should be the actual opposite. So there's no doubt that at some point something was going to happen to create a pandemic, whether it was from China, or whether it was from Russia, whether it was from the United States. We also found out that the United States was funding this whole operation in the United States until 2014, when it was then outlawed, basically, defunded. And what happened was that work got shifted over to the Wuhan lab, and the United States funds continued to flow where that research was over in Wuhan into a less safe, a less secure lab. So what was really going on over at Wuhan? Was Wuhan doing these experiments with the COVIDs in bats? Well, since 2004, shortly after the original SARS outbreak, remember that came from bats, researchers from the Wuhan lab have been collecting bat coronaviruses in an intensive search for a SARS-like pathogen. They wanted to continue with this process of these viruses that typically come out of bats. Since the original collecting trip, many more have been conducted. They were conducted in 2016, 17, and 18, or so it appears. So that group at the Wuhan lab has already performed experiments using those collected viruses. In 2013, the lab reported isolating an infectious clone of a bat coronavirus, and they gave it a localized name, the WIV1, which stands for the Wuhan Institute of Virology. And it was obtained by introducing a bat coronavirus into monkey cells, then passaging it. So we have proof that they were passaging things, these viruses from SARS. And they then tested its infectivity in human cells, engineered to express that human ACE2 receptor. There was a receptor in the human body that would accept this virus. So we know that they were working on gain of function, we know that they were working on passaging, and we know that they were working on the coronavirus. I don't know how much more circumstantial evidence and proof that we need to know that this was happening at the Wuhan lab in China. All the things that would create a potential pandemic if it was accidentally released or released on purpose, but more likely accidentally. Nobody's accusing them of having a bioterrorism act against the world. In 2014, just before the U.S. gain of function research ban went into effect, that Wuhan lab co-authored a paper with the lab from North Carolina that performed the original gain of function research on bat coronaviruses at the Department of Epidemiology at the University of North Carolina at Chapel Hill. In this particular set of experiments, the researchers combined the spike of the bat coronavirus. We all know the picture now from TV, the spikes, and they did it in a mouse-adapted SARS, what they call backbone, and they engineered it into a single live virus. And that spike was provided by that lab in China. They put this bat human mouse virus into cultured human airway cells and also into live mice. The researchers observed notable pathogenesis in the infected mice. The mice adapted part of this virus comes from a 2007 experiment in which that lab in North Carolina created the virus through passaging. So all these scientists were working on the same things to create a much stronger, more potent virus, and then how to make it such that it would become connected to humans through this passaging. Well, we don't know exactly what ultimately happened. And the only way to ultimately know is that in these labs, they keep very detailed records, because as they're doing all this research, they can develop concepts of intellectual property rights, of patents. And so these labs have to keep very detailed records. So if the virus was being studied, if gain of function was being performed, if passaging was being done, there would be notes in that Wuhan lab of exactly what happened and when and how they were studying it. That would give us great insight as to the source and the cause of the coronaviruses that the world is experiencing. But guess what? The Chinese government won't release those notes. And a lot of people question whether or not they still exist. They could have been burned and deleted because some of the lab researchers seem to be missing at this point who might be able to tell us. So there's a lot of shenanigans going on in these labs. So now what? Well, in 2019, the National Institute of Health lifted a 3-year moratorium on funding the gain of function research on the potential pandemic viruses such as the avian flu, SARS, and MERS, opening the door for certain types of research to resume. In a statement, the NIH Director Francis Collins said, "We have a responsibility to ensure the research and infectious agents is conducted responsibly, and that we consider the potential biosafety and biosecurity risk associated with such research." He added that he is confident that the new review process spelled out in the new framework will help to facilitate the safe, secure, and responsible conduct of this type of research in a manner that maximizes the benefits to the public health. They set up then a commission to develop a new set of guidelines, and it took several years for them to agree to these guidelines. And so when the Trump administration said we're going to start to fund the gain of function activities again, it was only after new procedures were put into place. So it wasn't like they said, well, let's go back and fund what was a problem before. We're just going to start sending dollars that way. That's not what happened. So they have a whole new panel called the National Science Advisory Board for Biosecurity. And they spent months hammering out this new process. This is very dangerous stuff. And so I think that we have to continue to be on guard. We have to have somebody that we can really trust in that scientific community. But it seems like it's going to continue to go forward. So what happens to all this research on the project that we were doing? That field work is not going to carry on in China because there's no funds for it. And that may be a good thing, because if we bring it home under more strict guidance, then maybe we can control these things that are supposed to be helping us, instead of turning around and biting us on the backside because we don't do it right. Well, what's the real source? When we wrap all this up, what do we say about the real source of the coronavirus? It still remains a question. Did it come from the Wuhan labs? There's 2 labs and people were going back and forth. The more likely answer is that it came from the Wuhan labs and was accidentally released. Did it just come from nature? Doesn't look like it, just like it didn't really come from the wet market and whatever might have happened at the wet market. It didn't come from nature. But we can't prove it one way or the other because the passaging hides the fact that it might have been developed and promoted in the lab. So what was the reason? Why would anybody do this? Was it an accidental release? That looks to be a more logical answer, that nobody would want to release something into the world. Was it a biological warfare? Unlikely. The real problem is what happened after the accidental release, if that's what happened. And that is that the Chinese government refused to let people understand and know early on what some of the impacts were as far as transmission from human to human. And the fact that the Chinese government went out and tried to get the protective equipment worldwide, get a monopoly on all that, and keep other people from getting it. They were not very open, as you would expect. They don't release the papers about what was actually happening in the Wuhan lab. So it looks like a massive cover-up by the Chinese government on an accidental release. That would be the best story. It's hard to believe that anybody, even the communist Chinese government that you can't trust and has been so belligerent with its own people, let alone others outside, that would release this as a biological warfare. So that's where we wind up. I hope you learned a few things today. Got a few new insights. None of it is trying to create a conspiracy theory. It's just trying to get the facts that have come from various academic papers, biology, research papers. I tried to look back at real details and facts and not try to drive a conclusion at the end that it was somebody's fault. We still don't know the answer to that, but the insights as to what was going on in that medical community, I think, is pretty fascinating, and hope we don't see any kind of accidental releases from this dangerous work that's going on. Well, join us again next week, and we'll find another topic to talk about on Healthcare Insight. You've been listening to America's Web Radio. I'm Ron Bachman. Until next week, we'll see you then.